Hi, I'm Larry Johnson. I am a professor at Texas A&M University, and today we're going to talk about epithelium. We want to talk about the embryologic origin and tissue organization of epithelium, the cell junctions and the polarity of cells of epithelium, how to classify epithelia, and the different types of glands and mechanism of glandular secretion. Thank you. Now, there's four basic types of the tissue of the body. So the whole body can be subdivided into four groups. One is epithelium, we'll talk about today. And since epithelium has a lot of regeneration power, they are about 90% of your tumors. There's connective tissue, muscle, and nerve, as we'll cover in the next few lectures. Now, remember when we talked about protoplasm, protoplasm is living substance, and the cell is the smallest unit of it. Some animals only have a single cell, but today we're talking about tissues, which are groups of cells with the same texture or the same function together makes a tissue. One of the four basic types of tissue, muscle, nerve, connective tissue, and today we're talking about epithelium. And if we look at the gut, we can see the lumen of the gut and also the outside of the gut, and both of those have an epithelial sheet. Indeed, epithelium is what makes cavities. So it makes the lumen, and also it makes the peritoneal cavity as well. That's what it does. And so that's epithelium in this case, but down here, since it's from a different origin, we call it mesothelium. Mesothelium surrounds the outside of the gut. And we can see it here, the epithelial cells that we've talked about so far with the brush border and all like that are really epithelial cells, and mesothelium are the little flattened cells that we see, or maybe cubes, sometimes columnar cells that are on the base. Again, what's in the blue, we see the intestinal or absorptive cells, which are epithelium, and we see the mesothelial cells as well. Now, epithelium, it covers organs, and it does that because it has a series of junctions we'll talk about a little later on. And those junctions allow cells to hold together and have little extracellular space. They line viscera, they also line blood vessels, and they are the secretory cells of the body, of our glands. So distinguishing features of epithelium is that they always sit on a basement membrane, and that come in a variety of conformations. They could have surface cells of different types. And if they're on one cell, we'll call it simple. If there's more than one, we'll call it striated. And they are attached to their neighbors, to each of the adjacent cells, by a series of junctions. It could be desmosomes, tight junctions, gap junctions, different types of junctions that we'll talk about. Now, if we're going to identify epithelium, we see simple squamous, that is one layer, and the cells are flattened. That's very good for lining blood vessels and covering organs. Also, it can have simple cuboidal. Cuboidal, namely because they're one layer and the cells are cube-shaped. Kidney, liver, many other ones. Simple columnar, that is, their cells are taller than they are wide. This is columnar with one layer. Or it could be stratified squamous. You always name by the cells on the top. These guys are always going to be cubes in the bottom. You name them by the top. Since this is flat like that, both being squamous, this is one layer simple, this being stratified as multiple layers, stratified squamous epithelium. And that's the type of epithelium that's protective. That's what you have on a callus on your skin, and that's what you have on the outside of your skin, as well as covering the cornea of your eye. Now, epithelia can also be pseudo-stratified as well. Namely, it looks like it's stratified, looks like cells are on top, but it's not. They all touch the bottom. They don't all touch the top, but they all touch the bottom. And we mostly find that in the respiratory system. Mostly they're ciliated, but you can also find them in the epidermis as well. And then there's transitional. Transitional is a surface cells could be more flattened, or they can be very fat in the case of relaxed state, and you find these in the urinary system. Now, epithelium can be specialized in different ways. It can have the surface amplified with the brush border, as we talked about before. It can have villi on it or cilia on there as well. And then also, in some cases, you have infolding of the plasma membrane, where you have outfoldings of the brush border, you have infoldings of these, and these line up the mitochondria. In doing so, it makes it striated. And so sometimes we see the striated. In the salivary gland, we even call them striated ducts. Now, in terms of origin, there are three basic germ layers all contribute to epithelium. On the outside, the ectoderm, it's just the epidermis of the skin. That's what we see in our neighbor. Epithelium covering the cornea, and those two together covers the entire surface of the body. Also, it lines body cavities. It covers. So from the ectoderm, it covers the surface of the body, and here come your sebaceous gland, mammary glands, tear ducts, various things you see from the ectoderm. There's also an endoderm on the inside. And in the endoderm, we have the tract, GI tract, and also from that come the liver, the pancreas, and also the endocrine organs. Now, they lose their connection to the surface, but still they come from the endoderm. And then the mesoderm is in the middle, and that gives rise to two types of epithelial cells, but they have their special name. Endothelium lines blood vessels, and mesothelium lines cavities, that we can say. So epithelium has specialization for functions, absorption, tall columnar cells, amplified surface area with a brush border. Secretion for the pancreas, pancreatic acinar cells, pyramid cells with lots of rough endoplasmic reticulum, and for making proteins. Transport in the eye, and in the endothelial cells, the blood vessels for transport. In the kidney, excretion and protection, that stratified squamous epithelium is right there, and also there's nerve endings in there that makes it painful. Ever scratch your cornea, you will know the pain. Also, these are the cells that are sensory. They sense pain. You have an injury, to avoid injury, and also they have taste buds and olfactory cells, a host of cells, and they contract, the myoepithelium. So here we see one of those cells, like a pancreatic acinar cell, as being the lumen up here, junctions making that lumen, rough endoplasmic reticulum, secretory granules to be discharged in the surface. Here we see a sweat gland, and in here we see an intercellular canaliculus. So this canaliculus right here is another surface area of the cell that's going to discharge into this big lumen up there. It's continuous. The lumen is continuous, that one. And you see, yep, there's a series of junctions around there that facilitate this cell attached to that cell. And then also we have some myoepithelial cells. Here it is. So these are epithelium, and these myoepithelium are epithelial cells as well. And so here we can see a host of sensory cells. There's a rod here in the eye, olfactory, this one in the ear, different ones that you can see. Okay, special surfaces area. You can have microvilli for the intestinal absorptive cell we've seen before. Could have cilia. This is the efferent duct in the male, which is a counterpart to the oviduct, but you can see the cilia projecting out through there. You see the basal bodies and the axoneme that would be projecting in through there. Also, their intercellular canaliculus, as I said, in hepatocyte, secretory canaliculus, and the parietal cells, we'll see in a minute, and the flagellum. And here you can see the flagellum of a tail that's developed, and germ cells, sperm cells, eggs, all those are epithelium. So to amplify the surface area, intestinal absorptive cell, we have a brush border, which are uniform microvilli of high density, and they're covered with a glycocalyx. And here we can see a parietal cell. This is a drawing of one, and this is a real one. And we see lots of mitochondria in through there, which is necessary to pump hydrogen ions out. But also you see that the lumen comes down deep within the cell. This is a secretory canaliculus that we're seeing through there, and you can see where it comes down deep in here. So the cell pumps out chloride, pumps out hydrogen ions. They get together and make hydrochloric acid, and the cell doesn't have to hold the hydrochloric acid. Inside is already in the lumen to be discharged in the lumen. In the hepatocytes, and here we see a series of hepatocytes. Not only do you have the lumen where the blood vessel is, you see here and there and in here, but also there's a bile canaliculus. Bile canaliculus, which is the intracellular canaliculus, and the hepatocyte. You can see it there, you see it there. And also you can see it here, where there's drawing showing you it. And this slide in this one shows you a staining for the bile canaliculus. So everywhere you have a hepatocyte, you're going to have a bile canaliculus. And in this H&E, we can not only see the nice nuclei in the cytoplasm, but you can also see the bile canaliculus reflectile here, there, bile canaliculus. And here we can see that epithelium can be specialized, could be flattened, as you can see. And so the endothelial cells are very special cells. They're not just sitting there sleeping. They have enzymes and receptors transport without much energy. They're flattened, so they have low turbulence. They have this marginal fold that flips over and helps protect the junction. And they're negatively charged, and that makes them non-wettable, kind of like they're waxed. So if you got fluid coming through, it's very handy if that fluid doesn't stick, if it moves along. And here we can see endothelial cell lining a valve. There it is right there again. These are fat cells, but these are in a lymphatic. So remember the transcytosis, where vesicle can move from one membrane domain to another membrane domain. Well, that's what happens in epithelium in terms of their secretory product. They make secretions in there, and the outside of the base of the cell picks up. It has receptors, the FC receptor, which binds to the IgA antibody, and then that takes it through by transcytosis and dumps it into there. So this is transcytosis of putting antibodies in the secretion. What secretions? Could be milk, tears, bile, seminal fluid, cervical fluid, etc. Body fluids have antibodies in there, and they use transcytosis to do so. So this is a modification of epithelial cells to accommodate, even though they have tight junctions here to keep luminal contents in here and not in the extracellular fluid, they have a way to pass good things through, which would be antibodies that they would have in through there. Now, specialization of epithelium, you can see there's a host of junctions in through there, little space between different ones, and these junctions make them polarized. They have a base, lateral border, and an apical border. And here we see one of those junctions up here, a tight junction, which is a fusion of the cell membrane from one cell and another cell that prevent things from coming through. And how many ridges you have dictates how tight of a junction it is. And it's not a spot well, it's a complete belt. That's what zonula means. There's also a zonular adherence, and this is another belt that goes all the way around this. And this one is the one that supports it, and it interacts with the actin filaments to help support it. This is just, zonular occlusion is just membranes that would pop. It prevents things from going through, but it's not stout. So you have to have zonular adherence just below. And then macular adherence are just spot wells in there. And then there's gap junctions, a uniform gap between two membranes, the membranes of two cells, that's how it got its name. And you can see there's also channels in through which information can go from one cell to another. And so here we see the cell with the junctions right up in this region. There's the zonular occlutants where you get a fusion of the membrane between this cell and that cell. Zonular adherence where you have actin filaments interacting, and then the macular adherence or the desmosomes you see throughout the connecting the cells. And here we can see it again, and we can see these structures in between. So this is one cell, another cell, right up here, and that is a terminal bars. The terminal bars that we can see there. However, if you cut this perpendicular to this, what you can see is the darkness of a terminal bar, which is really these junctions. It goes all the way around the cell. So indeed, it is a zone. So that bar goes all the way around the cell, not just one little thing there, and you can see the bar coming all the way around the cells, which are cut perpendicular right in this region. So zonular occludens prevent things from the lumen from going through, and the thing for extracellular fluid going into the lumen. And so here we see the zonular occludens. It prevents the channels that are in the apex from coming to the basolateral border. It restricts polarity. It restricts the movement, fluidity. It restricts the fluidity of proteins in the membrane that can't go down here. They can move around there. They can move around here, but these can't go there. And here we can see the tight junction of pancreatic aster cells, and lo and behold, here comes what would be the terminal bars, zonular occludens, zonular adherents, macular adherents. They're the same ones that make a lumen. That's how epithelial cells make a lumen. And if you look at the zonular occludens, you can see that there's many layers. 1, 2, 3, 4, 5, 6. Many layers here, and you can see where you get the fusion of the membranes between these adjacent ones, making this ridge that we see. The more layers we have there, the more of these fusions, then the tighter it would be. Now, the zonular adherence is associated with a belt of microfilaments. Microfilaments project up into the villi. They project all the way around the cell, and they're also a big band of them associated with the zonular adherence. The macular adherence also has filaments associated, but these are intermediate filaments. And they're attached to desmosomes and the hemodesmosome, which is a half a desmosome. So both the intermediate filaments and actin filaments are responsible for structural support inside the epithelial cells. And here we can see the gap junction, uniform gap, that is uniform space between this cell and that cell, and you can see the gap junctions that facilitate communication. Here you can see the little holes in the gap junction cut perpendicular to the surface of the thing. And here you can see little holes. And a certain molecular weight, 1,000 molecular weight and less, can go through. Larger ones cannot go through, and those things have to come through with a different mechanism. So if we look at the tight junctions, mostly it is a fusion of the membranes, but it needs to be located at a certain place. Below that, you've got the zonular adherence, which has actin filaments, and then a desmosome, hemodesmosomes, we get intermediate filaments, and these are the keratin filaments, and then we have gap junctions throughout the cell. Now you can have, since actin filaments is associated with the surface of a plasma membrane, shape of the membrane, it's also associated with the tight junction, probably to keep it in its location. So I mentioned there's junctions holding cells together, but that's not the only proteins. There's other proteins, integens, and catechins, selectins, and all these also help bind cells together. Not to be concerned about it in our class, just to know that what we're saying is just the tip of the iceberg in terms of the cellular connection between and communication between the cells. Now you classify epithelium by one layer or more. One layer is simple, more stratified. And then the surface cell. Are they flat? That's squamous. Cuboidal. Columnar. Columnar has more cytoplasm above the nucleus than they do between nuclei. And so that's where you see that. Also, the nuclei tend to be more oval as opposed to spherical because that's the shape of the cell. And then there's other things. There's pseudostratified, as I mentioned, and transitional. Those are the two. So if we look at blood vessels, lining blood vessels, we see endothelial cells. They're flattened, simple, squamous. And then we have simple cube cells in the kidney and in the surface of the lens of the eye. Simple cubes, simple cuboidal. And then we have one layer of columnar cells in the gallbladder and intestinal absorptive cells. We can see. So there's this one cell, it's a column, another column, another column, series of simple cuboidal, but columnar, because they don't have but one layer. Then we have stratified squamous. So the flattened cells on the surface, and they can be keratinized, as we see in skin where you touch the air, or non-keratinized like you see in the cornea of the eye. So stratified squamous, this is what we see. These are the layers. They have division of cells right between the epidermis-dermis junction, and these cells ultimately are sloughed off at the top. And we can see desmosomal connections connecting. You can see here and here as cells are connected by the desmosomes. So this is epithelium, connective tissue down below. Stratified squamous epithelium, that was. A stratified cuboidal, as we see, only know that in the sweat gland ducts. So the more than one layer, one sits on the other one, and they're mostly cube cells. There's stratified columnar in the male reproductive urethra, more than one layer, and cells are columnar on the top. Stratified columnar is in the epidermis as well as the respiratory system. And transitional could be like these big fat cells on the surface in the relaxed state, or they can look almost squamous whenever they are extended out. The cell membrane doesn't stretch, it unfolds. It's transitional in the urinary system. So in summary, it's avascular. Epithelium does not have blood vessels. It gets its nourishment from the connective tissue below. Extraneous cells, you always have lymphocytes and white blood cells migrating through. Regeneration ability, you have amazing ability to regenerate cells, and anytime you got a rapid regeneration, you have opportunity for cancer, and that's why about 90% or so is epithelial cells. Not only because there are many epithelial cells of different types throughout the body, so that would also increase the likelihood of being cancers too. Also, migration. Cells can migrate from one layer, from one location to another. If you ever scratch your cornea today, it's painful, but tomorrow it'll be covered over. Not because new cells, because those have slipped and slide. If you have a burn, the degree of burn depends upon how far you go down. If you go down below your sweat glands, then you're going to have a transplant. But if you got sweat glands still down in there, then they can repopulate the surface because they regenerate, and then also because they migrate up. Metaplasia is where they can transform to another type. And when they transform to stratified squamous epithelium. That's what they do in the respiratory system, and that's what it does in other places. Basement membrane. They all sit on the basement membrane. Remember, the basement membrane is what you see at the light microscopic level. What we see at electron microscopic level is a basal lamina. The basal lamina is part of the basement membrane, okay? But the basal lamina is not as big as the basement membrane. So secretion of cells. So we have endocrine cells and we have exocrine. In the case of exocrine cells, they're developed and they stay attached to the gland, to the ducts. So they discharge on some surface. Okay. In contrast, the endocrine cells lose their attachment to the surface, lose it. And as a consequence, they discharge their secretions in the connective tissue, which ultimately gets into the bloodstream. So there's three basic types of secretion. One is Merocrine, which is exocytosis, without loss of the surface. And remember, this is what it was here. Exocytosis, what we did in the pancreas, we talked about it. Sometimes the proteins are stuck in the membrane, other times exocytosis. And that's Merocrine. All the cells would have to use that. Apicrine, where some of the cytoplasm actually comes out with a secretion. So the apical cytoplasm comes out. Lost part of the plasma membrane. Holocrine secretion, whole thing goes, like a sebaceous gland. And then cytocrine, where certain things are produced by one cell just for another cell. And that's what we have in our skin. Merocrine secretion and milk secretion, casein comes out there. Other things come out that way. But apicrine is where you have the milk. Milk is fat, milk fat, not only milk, but fat. Fat is not water soluble, and so you have to have a membrane around it so it can be solubilized, is what it is. So the lipid droplet coming through is surrounded by the cell membrane in the apicrine secretion. And so we have the Merocrine secretion in the thyroid, and it creates this follicle full of a substance in the middle, colloid. That colloid there is secreted, and then it's phagocytized, broken down, and then by Merocrine secretion, you discharge protein hormones, as you see. And so if you're outside and stress goes to the brain, comes to the adrenal, you open a door and see someone's there, and then so here it comes to the adrenal, here comes epinephrine, and your heart sinks. That's what happens. In this case, it's causing glucose to be discharged. In this case, it was doing the endocrine. And the endocrine is where signal comes from one place, and then another cell reacts to that signal, which is carried through the bloodstream. Adrenal, kidney, liver. And then we have the exocrine gland, different ones. If it's unbranched, we call it simple. If it's branched, we call it compound. So this is the duct portion. There's also a secretory portion. Sometimes it can be a tube. Sometimes a coil tube. Sometimes branch tube. Sometimes alveolus as opposed to acestus, branched acestus, tubular acinar, tubular alveolar, and different combinations you can have. And then also there's serous and mucus. Serous is a proteinaceous, and mucus is like more carbohydrate type mucus. Flat nucleus at the bottom and secretions in the apex, as opposed to here you got a spherical nucleus pretty much in the middle and very eosinophilic. And so if we look at a goblet cell with the secretions out through there with Merocrine secretion would be what we see in the goblet cell is really the theca. The theca here is a secretory part, but it does have a nucleus as we see there. So in terms of the duct, the gland is the duct part branched or not? None of these are branched, so they are all simple. And it could be a simple tube, a simple coil tube. It could be a simple branch tube. It could be branched alveolar, branch tubular, different ones that you can have. Branch acinar, tubular different ones you can have. And so here we see the tubes branch. That makes it compound. And so this is a compound acinar, tubular acinar, different combinations that you have. And these ducts are named different ones: interlobular, intralobular, intercalated. They're different ducts. Don't worry about them right now, but we'll get them later on. And here we can see where you have a serous demilume, where you have a serous on the outside and a mucus on the inside, and there's little channels where this fluid goes into there, and that's called the intercellular canaliculus that we talked about before. So it's a serous demilume. Proteinaceous part carbohydrate part here. So in summary, epithelium comes from all three germ layers. If it's from the mesoderm, they have a special name, mesothelium and epithelium, and endothelium. And then so the cells could be flattened, could be cubed, could be columnar. They could be one layer, they could be multiple layers. Now this is something you need to look at real carefully, because the lateral border is and basal border is different from the apical border. And so you'll need to name 3 or 4 things which are different in the apical and lateral border. Not only what's different, but also what's the function there as well. So here we can see there's junctions on the sides. As we can see, you have direct transport, separate, indirect sorting. You have proteins that are there. And when we just look at our intestinal absorptive cell, we can see that we have symports here, symports, and then uniports down here. So these are different than those. So I just told you 2 things that were different between the apical and the lateral border. And the function is whether glucose is going against this gradient or down its gradient. Now there's several books that we have taken illustrations from. We thank them for the contribution to this presentation. They made those. The credit is theirs, not mine. I just took pictures out of these books to use and organize them from the different books, and we thank them. Especially right now, we're still talking about Albert's books, but also other books as well. Next time, we're going to talk about connective tissue. And what does connective tissue do? Yes, it connects. Either that or we're going to go to Tokyo. Here's Tokyo, Japan. Here's our bullet train. Yes, it is an electrical train, and it is fast. And the guy told me, just pick the blue one. How do I know which bicycle? Look at all the bicycles. These are people there. Look at all the bicycles that are there. How on earth can you ever find your bicycle once you get back in there? I can't imagine how you find your bicycle. And then also Mount Fuji. Mount Fuji, we rode a bus all the way up to the snow line. Is what we did. And also walking down the street, I saw a little boy with a T-shirt on from Texas A&M. And when I saw that, I got so excited. His mother thought I was going to kidnap the boy. I had to step back with hands up, saying, no, I was not going to do that. I gave her my card, which was on one side Japanese, and lo and behold, that little boy's granddad, her dad, had come to Texas A&M University. This was right outside of Tokyo in a little town called Atami. Thank you.